My name is Zoe Davis, and I'm a graduate student here at Berkeley in Britt Glansinger's lab. And today I'm going to tell you a bit about some of the aspects of late gene expression that we've been studying. So in the Glansinger lab, we study Kaposi sarcoma-associated herpes virus, or KSHV. And KSHV causes B-cell lymphoproliferative disorder. Thank you. So KSHV causes B-cell lymphoproliferative disorders, including endemic and HIV-associated Kaposi sarcoma. And in untreated AIDS patients, KSHV still is the leading cause of cancer, and it remains a major cause of cancer in Africa. KSHV is a large double-stranded DNA virus. It's about 130 kilobases and encodes for about 90 genes, all of which are expressed during the productive or lytic phase of the viral life cycle. And as with all herpes viruses, the genes are expressed in an ordered cascade, beginning with immediate early and delayed early genes, which are things that are usually involved in modulation of the host response, and some of those products are also necessary for the next step of the viral gene expression cycle, which is the replication of the viral genome. Now, only after you get replication of the viral genome do you license the next step of viral gene expression, which is the expression of late genes. And this relationship between DNA replication and late gene expression is one that's conserved through all DNA viruses and not really very well understood. So late genes are typically, their protein products are structural in nature and are required for the formation of progeny virions. So late gene expression actually kind of poses some interesting challenges because late genes, once they need to be very tightly regulated, only turned on at an appropriate time. And when they are turned on, you need large amounts of them produced. But the time when they're needed coincides with a period of significant stress for the cell. So trying to understand how the cell can actually coordinate this and actually orchestrate this regulation has kind of been a mystery. So though late gene expression has been kind of a black box, some things are known. As I mentioned, there's this absolute requirement of late gene expression for replication of the viral genome. What's also known is that the promoters actually have atypical TATA boxes, which instead of containing the classical TA-TA core motif, actually is comprised of TATT. And additionally, the promoters are actually quite minimal, consisting of little more than the TATA box. And it's quite striking that you have all of these, this kind of minimal promoter, yet you still get this very precise regulation. So previous work in other herpes viruses have identified 5 viral proteins that appear to be essential for only for late gene expression. So what that means is that when these are deleted, they don't affect any of the earlier parts of the gene expression cascade. They only affect late gene expression. So we were very interested in trying to understand how these proteins influence late gene expression and what host machinery is actually being used in order for this to occur. And we focused our studies on one of these 5 proteins, which is ORF24. And ORF24 was an interesting place to start looking at late gene expression because several years back, the Rychlewski group published a genome-wide fold recognition search looking for proteins that might be able to bind the unusual TATT motif found in late gene promoters. And what they came out with was the ortholog for ORF24 from Epstein-Barr virus. And they further predicted that this domain, that ORF24 ortholog contained a domain that actually folded like TATA box binding protein, or TBP, despite having very low sequence identity. So just to remind you, TBP, along with the TBP-associated factors, or TAFs, comprise TF2D, and TF2D binds to the TATA box and then serves as a kind of a nucleation, nucleates the recruitment of other general transcription factors, ultimately recruiting the polymerase and then leading to the initiation of transcription. So what we thought is that perhaps this TBP-like domain could be contributing to the recognition and the transcription of viral late genes. And in fact, it was shown with the ortholog in Epstein-Barr virus that, that it was actually able to bind late gene promoters. So what we next did is we looked based on the predicted similarity of ORF24 and TBP, we predicted residues that might be able to, that might be contacting DNA, and then made targeted mutants to look at those. So what we found is that ORF24 is able to bind late gene promoter DNA, which you can see here from this gel shift. And then when we look at, do the same experiment, but using versions of ORF24 where we've mutated the residues that we predicted from this TBP homology to bind DNA, we see that we lose that shift. So then we wanted to know, okay, so what effect does this actually have on late gene expression? And what the way that we address this is we made a mutant version of KSHV where it does not express ORF24. And then we added back the mutant versions of ORF24 and looked to see how that affected late gene expression compared to wild type ORF24. And what you see is that there's a defect in late gene expression and that these mutants do not rescue it while wild type does. So what this kind of suggested to us is that that ORF24 is clearly binding this promoter DNA, and that this is necessary to get full activation of late gene expression. And in data I don't have time to show you today, we also further showed that ORF24 binds at late gene promoters in place of TBP, which is kind of an interesting thing when you think about how important TBP is for transcription and for kind of nucleating the recruitment of all of these other factors. Yet we don't really understand the mechanism, but we know that other general transcription factors are being brought into the transcription pre-initiation complex, but we don't fully understand how that is working at this point. So we know that ORF24 is binding the TATT motif at late gene promoters, but once it's there, how is it actually facilitating transcription? So for that, we turn to our data, our human KSHV interactome data for ORF24, which we did in collaboration with Nevin Krogan at UCSF. And looking at the data for ORF24, a complex clearly stands out, which is that here of RNA polymerase II, the polymerase responsible for making mRNA. And we were able to confirm this interaction by transient transfection as well as by inserting a flag tag into the KSHV genome on ORF24 and then doing IPs here for ORF24 and then blotting for the large subunit of POL2. So I further fine mapped the interaction site and I was able to narrow down the interaction between ORF24 and the polymerase to 3 leucines that are found in the amino terminal domain of ORF24, and these are highly conserved through orthologs of ORF24. So we then used these mutants that are selectively defective in binding POL2, they otherwise seem fine, and looked to see if they were able to facilitate late gene expression in normal levels using the same assay as before. And you can see, as with the TBP-like mutants, they're not able to complement late gene expression. So then we have a model here where we have ORF24 kind of has 2 genetically separable functions in that it is binding late gene promoters and also binding the polymerase. It seems to bind in place of TBP, and that both of these functions are required for late gene expression. So kind of on a more speculative front, what we see some similarities here to the strategy used by prokaryotes in using sigma factors for transcription initiation. And the similarities we see is that this is highly specific and it induces robust transcription of its specific class of genes. And the obvious similarity is also that it binds directly to the DNA while also recruiting binding the polymerase. So we wonder about why this strategy might be advantageous for the virus. And one of the things that we've considered is that at late times during infection, TBP levels actually go down, the protein levels go down, because the TBP mRNA is actually a target of the viral host shutoff factor, SOX. In contrast, ORF24 mRNA is not a target of SOX. It's spared from degradation. Thus, its levels kind of stay constant and are fine. So at late times in infection, when TBP levels are low, you need to have late gene expression. So perhaps the virus found this strategy, happened upon this strategy in order to facilitate expression of late genes at this time. So with that, I'd like to thank our funding sources, and especially SEND, and our collaborators in the Krogan lab. And I'd like to thank Britt and all of the members of the Glansinger lab. And I can take any questions.
That was a beautiful talk. So is the binding of the ORF24 specific to the TATT box, or can it also bind to a regular TATA box and screw up cellular genes, basically?
So we don't fully know that. In vitro, just by gel shift, we can get it to bind. If we take like a same probe that has a TATT with a late gene, and we just take that last T and get back to an A, it will still bind. If we move, if we change like all of the residues to Cs, it won't bind. We are very interested in trying to identify if it has any effect on cellular genes. That is definitely on the to-do list. I think it's a really cool idea.
So it looked like there was still some ORF52 mRNA being produced. Does your ORF24 mutation completely prevent case HV replication, or just partially?
So when we look at protein levels, we can't detect any protein of these late genes. And then when we look to see if we're producing any virus, we're not producing any virus. So our guess is that from the qPCR that we're kind of seeing some residual, we have a lot of overlapping transcripts, and that we might be kind of detecting something like that. As far as we can tell, late gene expression seems to be pretty thoroughly gone.
Hello. I'm not sure if I remember this totally right, but I think in human cells there's also some homologs of TBP, like TRF2 and 3 or so. Mm-hmm. And I'm not quite sure. I mean, is that something similar to what has happening here in the virus, that they use it for certain?
Yeah, so that's a great point. So those homologs, they have a lot of sequence similarity with TBP, and what's unusual about this is we have extremely low sequence identity, and that kind of makes an issue for how are you going to recruit all of these other factors to bind. It is also an issue because the promoter is so minimal, we don't even have sites for all of these other things necessarily to bind. We think that our initial chip experiments would indicate that we're getting at least some of the general transcription factors brought in, but we haven't looked to see if there's any of those kind of TBP-like proteins that are there, but from our mass spec data, it doesn't suggest that ORF24 interacts with any of those.
So our last question back.
Yep. My question is regarding this motif that's bound by ORF24. Do you know of any? Have you looked whether there's cellular genes that are upregulated, that are ORF24 dependent?
So we haven't looked at that yet, but I think that is a very, very interesting thing to look at. You could imagine a scenario where herpes viruses are ancient, and that there's kind of some genes that it would be advantageous at late times in infection for those to go up. But yeah, at this point we don't know. It's definitely something we'd like to look into.
Okay, thank you.